Hi, in this section, we're just going to hit some of the highlights that go with the cell cycle, mitosis, and meiosis, so that you can see how these things are going to relate to the formation of gametes and fertilization. So the first thing that we have is we have replication or duplication of the genetic material. So after the long strand has condensed down to form a visible structure, we call that structure a chromosome. And we have homologous chromosomes for all of our pairs of chromosomes except of our pairs of autosomes. So we have a short arm and a long arm attached in the middle and the centromere, and you've gotten one from one parent and one from the other parent. So in the process of duplication, we go from this type of a chromosome to a chromosome that looks like an X. And the X shows you that the chromosome has been duplicated as far as the amount of genetic material. And each part of the X is called a sister chromosome. So the confusion is, after duplication, we still call it a chromosome with sister chromatids. Before duplication, we just call it a chromosome. So it's weird in that we both call them chromosomes. So what is mitosis? Well, in mitosis, we have that one duplication where we have that chromosome, then we make the chromosome with the sister chromatids. And after that one duplication, we have one division. And so we end up with those sister chromatids separating from each other, one going to each daughter cell. So they're always called daughter cells, whether it's in males or female. Okay. So because we are taking the genetic information and duplicating it exactly, that means both sister chromatids are exactly the same. And so in the daughter cells, each of them has the exact same duplicated genetic material as the parents, so they're identical to each other as well as to the parents. So that's mitosis in a nutshell. In order to do this, there are multiple phases. When we have not entered mitosis for the rest of the cell cycle, we are in the part called interphase, where you clearly see a nucleus. In prophase, the nuclear membrane has disappeared and the chromosomes have started condensing and they are visible. After that, they line up in the center and you can see the spindle formation coming from the centrioles. In the metaphase, for in the middle. When they pull apart, that is anaphase. And then telophase is basically the reverse of prophase, where the chromosomes are going to get all mixed up again, and then they're going to decondense, and you're going to have a new formation of the nuclear membrane. And cytokinesis is when the cytoskeleton of the cell causes the cell to divide in two. So that's great, but that's not what happens when you want to have offspring. For that, we need a second process known as meiosis. And in this one, we're going to call the daughter cells gametes. So what has to happen in meiosis is we need to start with a lesser number of chromosomes. In mitosis, we say the cell has 2n, okay? Where n's the number of chromosomes, and it's 2 because you have one each from mom and dad. Whereas in meiosis, we are going to have half the amount of genetic material. So we are going to be what's called a haploid cells, or they're also called cells that have N. And so when these two cells, the oocyte and the spermatocyte that are haploid meet together and fertilize, then you end up with a diploid or 2N cell that undergoes mitosis for growth and development. So this is why we need another process. So how does this occur? Well, we can't just get rid of, say, all right, so I know we have got this many pairs of chromosomes and we're just going to get rid of 12 of the pairs. It's not the way it happens. What happens is we have our one duplication where we go from all 46 of our chromosomes, where we have 46 sets of sister chromatids. And then instead of lining up single file in mitosis where the pairs get separated, the sister chromatids get separated, what happens is they line up where, for instance, here's chromosome 1, sister chromatids from mom, and there's chromosome 1, sister chromatids from dad, and they line up on what we call tetrads in the middle. So we're going to have two divisions. The first division divides the tetrads, and then the second division divides the sister chromatids. So we end up going from 2N and ending up with 4 cells at the end that are haploid. The first division takes us from 1 cell to 2 cells, and the second division takes us from 2 cells to 4 cells. So how we can tell which phase we're in is we use the same words: prophase, metaphase, anaphase, telophase, and we just stick a 1 in front of it if we were talking about the first meiotic division, or meiosis 1, and we put a 2 behind it if we're talking about meiosis 2. So in the beginning, we have this homologous pair of chromosomes, and this particular orange is coming from the dad and red's coming from the mom. And so we are going to have our 1 duplication, and now we still have homologous pairs of chromosomes, but each one has a pair of sister chromatids. So this is what is happening in prophase. Now, what happens is when they line up in their little tetrad 4 across, what happens is mommy and daddy play footsies under the table there. Okay, so if you look at the long arms, what you will see is something called crossing over. And what happens is a portion of the long arm of each breaks off and attaches to the other homologous chromosome. So we have this exchange of genetic material. Some of the genes that you got from your mom are now on a chromosome that you got from your dad, and vice versa. So now we can tell when we look at the sister chromatids at the end of prophase 1, we can tell that we're in meiosis because this process does not happen in mitosis. Now, there is a second thing that happens in meiosis that doesn't happen in mitosis, and that is something called random alignment. And this happens in metaphase 1. So what this means is, remember, in meiosis, the chromatids, homologous chromosomes, line up side by side. So we have a tetrad, whereas in mitosis, they lined up single file. So when they line up in tetrads, we could have a mom chromosome over here and a dad one over there, or we could put the mom one over here and the dad one over there. And we can do that for all of our pairs of chromosomes. And so this is a random lining up, or what's called independent assortment. So it's not like kindergarten where you had 1 line of boys and 1 line of girls. You still have 2 across, but they can line up however they want. And so if we look at those 23 chromosome pairs, we could have 23 lined up exactly, dad's over here and mom's over there. Or we could have it 22 and 1, or we could have it 21 and 2, or we could have this, as you can see, all the way down. And that gives us more than 8 million different combinations, which means that the gametes are not going to be the same. This is why you don't look like your siblings. Well, it's one reason why you don't look like your siblings. And that crossing over is another reason. Because what is wrong with this picture is not showing the evidence of crossing over, which has actually already occurred. So the effects of random alignment, if we just take that without showing evidence of the crossing over, possibility 1, if we only had 2 sets of chromosomes and all the dads are on one side, all the moms are on the other side, when they divided down the middle, when you were in meiosis 2, you would end up with 1 chromosome, 1 cell that had all of dad's genetic material and 1 that had all of mom's genetic material. So in your final 4 cells, half would be just dad stuff and half would be just mom stuff. If we still stick with 2 chromosomes, but we flip them so that 1 pair has moms on the left and dads on the right, then when it divides down the middle and you have 2 cells, you realize you have 1 chromosome of each. And so when the daughter cells divide, all the chromosomes have both genetic material from mom and dad. And this one, 2 cells have genetic material from the blue chromosome, long one of dad, whereas that same chromosome of mom is in the other 2 cells. But then on the second chromosome, the one that had dad's material previously now has mom's, and vice versa. So, summarizing meiosis. We start with a single chromosome, we duplicate the material, that's done in interphase. Then in meiosis 1, we have prophase 1 with the condensation. We have crossing over that is occurring. We have evidence of crossing over that occurs. We also had the independent alignment. The crossing over occurred way up there in prophase, in case you didn't notice it. We have the independent assortment or random alignment, and so we have mixed chromosomes. We end up with 2 cells at the end of meiosis 1. And then in meiosis 2 and prophase, those chromosomes line up single file, and now we separate the sister chromatids. And so if we look at these final cells, what we will notice is we have 4 cells. They're all haploid. And if you look, you can see evidence of crossing over in some of these cells. So, mitosis versus meiosis. Mitosis, 1 duplication, 1 division. Siblings look like each other and look like the parent. Meiosis, 1 duplication, then 2 divisions. Plus we've had 2 things which lead us to genetic variability. We've had crossing over, and we've had the random alignment / independent assortment, whichever term you want to use it. And because of the 2 divisions, we end up with 4 haploid cells. So at this point, pause the slide. This shows evidence of everything that is happening, and talk yourself through this process of mitosis on the left versus meiosis on the right. Okay, and there is your summary, mitosis versus meiosis. All right, and with that, I am done with the highlights. If you have any questions, watch this again, or watch the crash course videos, or ask questions. And I will see you again soon. Thank you.